Welcome back to What Did I Do in Lab last week, where I tell you guys what I did in my biochem lab last week. So this was actually the last week of lab for the semester, so this is the last video on this, but this week was my ELISA assay lab. So what ELISA is, it's a enzyme-linked immunoassay. So what it basically does is it detects certain antigens of an infection or antibodies against those antigens of an infection. So what we did, me, my lab partner, and everybody else in the class had to go grab a tube, and it had a sample of stuff in it, and that stuff was either positive or negative for COVID. So it wasn't actually COVID, that was just the example we were using in lab. So we grab the samples, and then what you do, start off with you and your lab partner, and you mix those two samples together and then split them up again, so we each have the same amount of liquid, but they're just mixed now. And then you go find another group in the class and you give them some of your body fluid and they give you some of their body fluid, and you exchange that. And then you do that again a third time. So you've exchanged with your partner, one person in the class, and another person in the class. Then what you do is you perform the ELISA assay, where you basically just put a bunch of stuff into these little plastic thingies. So there's these little plastic things, and you put a bunch of stuff in there. There's a positive control, a negative control, and then the sample of my fluids and my lab partner's fluids. Then you add a bunch of stuff, and it basically detects the, in this case, COVID spike protein, but not actually. And then if it's positive, it turns blue, and if it's negative, it stays clear. So, and then, since the entire class exchanged fluids, there was only one sample that was positive initially, so we were actually able to track where the infection originated by seeing who ended up positive and who ended up negative. Overall, my lab partner and I both ended up negative, so even if a person we exchanged with ended up positive at the end, doesn't necessarily mean that we were positive, just that that person exchanged fluids with somebody who was positive later. So since my lab partner and I both ended up negative, as you can see, and I think it's the far right one there, that means all the people we exchanged with ended up negative. And this is the same principle of how rapid tests work. Like, you know, when you take the rap test and it shows up there's lines on the thing. This is the same principle, and it's very useful for tracking how infectious disease outbreaks move. And it's interesting.